Hello students, let us start with part 7 of chapter 3, Inheritance and Variation. In the previous part, we have studied the structure of sex chromosomes, the concept of linkage and crossing over, Morgan's experiments on linkage and crossing over, and autosomal inheritance with the examples of widow's peak and phenylketonuria. Let us start with the sex-linked inheritance. Genes, those are located on non-homologous region of sex chromosomes, are called sex-linked genes. The traits or characters that are determined by sex-linked genes are called sex-linked traits. The inheritance of sex-linked genes from parent to their offsprings is called sex-linked inheritance. Two types of sex-linked genes as X-linked genes and Y-linked genes. X-linked, they are also called as sex-linked genes. X-linked genes are located on non-homologous region of X chromosome, and these genes do not have corresponding alleles on Y chromosome. As female has two X chromosomes, in a female, two recessive sex-linked genes are required for expression of sex-linked traits. If one X chromosome carries a recessive gene for sex-linked trait or any defect, its effect is suppressed by the dominant gene present on other X chromosome. The females with only one recessive gene are called carriers. The carrier female is physically normal in appearance, as she does not suffer from the disease or disorder. Males have only one X chromosome. If X chromosome carries X-linked recessive gene for sex-linked trait, then it is expressed phenotypically because there is no dominant gene present on Y chromosome to suppress its effects. Therefore, students, the sex-linked or X-linked traits appear more frequently in males than in females. Examples of X-linked traits includes haemophilia, colorblindness, night blindness, myopia or short-sightedness, and muscular dystrophy, etc. Second type are Y-linked, or they are also called Hollandric genes. Genes which are located on non-homologous region of Y chromosome are called Y-linked genes. The Y-linked genes are inherited directly from male to male. Example is hypertrichosis. Hypertrichosis is responsible for excessive development of hair on pinna of ear. This character is transmitted directly from father to son. In the diagram you can see the presence of hair tuft on pinna of ear is called hypertrichosis. Let us start with the inheritance of colorblindness. Colorblindness is X-linked recessive disorder where person is unable to distinguish between red and green colors, as both the colors appears grey to him. In this diagram you can see the first diagram shows the vision by a normal person, and the opposite diagram shows the vision due to the colorblind person. It is caused due to recessive X-linked genes, capital X, capital C, which prevents formation of color sensitive cells called the cone cells present in the retina of eye. Homozygous recessive females having genotype capital X small c, capital X small c, and hemizygous recessive, that is capital X small c y, are unable to distinguish between red and green colors, so that these are the colorblind persons. The frequency of colorblind women is much less than the colorblind men. Dominant X-linked gene represented by capital X, capital C is necessary for formation of color sensitive, that is cone cells in the retina of eye, which gives distinguish for the green and red colors. The genotypes of male and female individuals can be represented as follows. So, the male who are normal, that is having normal vision, have genotype X capital C Y. Those who are colorblind shows genotype capital X small c Y. Carrier males are not there. Female which have a normal vision have a genotype X capital C X capital C. Those who are colorblind shows the X-linked recessive gene on both the chromosomes, hence they have a genotype X small c X small c. And the carrier female who is phenotypically normal but shows a gene on one of its X chromosome is represented by genotype X capital C X small c. The inheritance of color blindness can be studied in the following two types of marriages. Consider the first marriage. It is a marriage between colorblind male and normal female. Will produce normal visioned male and female offspring in F1. You can see a chart where the parents in which colorblind male having genotype X small c Y when married with normal female having genotype X capital C X capital C. They produces gametes X small c Y X capital C X capital C. When the gamete having X small c fused with the another gamete from female X capital C, it produces the genotype X capital C X small c. Carrier daughter. When X small c fuses with the another gamete from the normal female X capital C, it again produces a carrier daughter. When the Y fuses with X capital C, it produces a normal son. Again the one Y, when it fuses with the another gamete from normal female X capital C, it produces normal son. That means the sons, they have a normal vision, but the daughter will be carrier for the disease. 50% are the carrier daughters and 50% normal sons are produced by this type of marriage. Second, the marriage between the carrier female and normal male. So, the carrier female or daughter shows genotype capital X capital C X small c and normal male X capital C Y. When the one gamete from carrier daughter X capital C fuses with the one gamete from the normal male X capital C produces normal female 25%. When again the same gamete X capital C fuses with the Y, normal male is produced 25%. When the gamete from carrier daughter X small c fuses with the gamete from the normal male X capital C produces carrier daughter having genotype X capital C X small c 25%. And when the one gamete, that is X small c fuses with the Y chromosome, it produces colorblind male having genotype X small c Y 25%. That means This marriage will produces female offsprings which are carrier but with normal vision, and half of them will be the carrier for the disease. Half of the male offspring will be normal, while remaining half will be colorblind. That means this says that the inheritance of colorblindness is in criss-cross manner. That means the colorblind male passes the genes for colorblindness to the female, and from the female through the carrier daughter they are again passed to the male. This type of inheritance is called as criss-cross inheritance. X-linked recessive genes for colorblindness is inherited from colorblind father to his grandson through his daughter. Criss-cross inheritance. Second example of criss-cross inheritance is haemophilia disease, which is also called as bleeder's disease. Haemophilia is again an example of X-linked recessive disorder in which blood fails to clot or coagulates very slowly. The genes for normal clotting are dominant over the recessive genes for haemophilia. The person having recessive gene for haemophilia is deficient in clotting factors, that is factor number 8 and or 9 in blood. Even in minor injuries causes continuous bleeding in these persons. Hence, the haemophilia is also called bleeder's disease. The recessive genes for haemophilia is located on non-homologous region of X chromosome. As there is no corresponding alleles on Y chromosome to suppress its expression, so men suffer from this disease, same as like the color blindness. X chromosome have recessive alleles. The genotypes for male and female individuals can be represented in the following chart. The male who are normal have the genotype X capital HY. Capital H represents the normal or dominant gene. Hemophilic male have genotype as X small hy. Carrier males are not there. Female who are normal have genotype X capital H, X capital H. While hemophilic have genotype X small h, X small h. And carrier have the genotype X capital H, X small h. Let us see the inheritance of the hemophilia. Like color blindness, hemophilia also shows criss-cross inheritance. The inheritance of hemophilia can be studied with the help of following examples. The first, marriage between hemophilic male and normal female. Hemophilic male have genotype X small hy. Married with the normal female having genotype X capital H, X capital H. Hemophilic male produces two types of gametes, X small h and Y. Normal female produces same kinds of gametes, X capital H, X capital H. When the gamete having the X small h fuses with the gamete from normal female X capital H, it produces carrier daughter. Again the same gamete X small h, when it fuses with the another gamete from normal female X capital H, it again produces carrier daughters. Carrier daughters are produced 50%. Then Y gamete from hemophilic male, when it fuses with the gamete from normal female X capital H, it produces normal son. Again the same Y gamete when fuses with the another gamete from normal female X capital H produces the normal sons. So the carrier daughters and normal sons are produced 50-50 in percent. In the second marriage, between carrier female or daughter and normal male. Carrier daughter having genotype X capital H, X small h. When it married with the normal male X capital HY, the offsprings which are produced are normal female 25 in percent, normal male again 25 in percent, carrier female 25 in percent, and the hemophilic male they are 25 in percent. Here the colorblind is written instead of hemophilic. The inheritance is same as like the color blindness. Students, do you know the hemophilia is also referred as the royal disease because it affected the royal families of England, Germany, Russia and Spain in 19th and 20th centuries. The Queen Victoria of England, who ruled from 1837 to 1901, was believed to have been the carrier of hemophilia. She passed the trait on to her 3 of 9 children. Queen Victoria was the first known carrier of the blood disease haemophilia, which would become known as a royal disease. Okay. Students, in the next part we will study the sex determination in human beings, birds, honeybee and genetic disorders. Thank you.